Hi guys. So I'm recording this quick video because our lecture got canceled due to the power outage, and I want you to have an opportunity to see this cancer material since we didn't get a chance to talk about it. So my goal for the end of this little lecture is that you'll be able to talk about the aberrant cell division, or when cell division goes wrong, and the regulation of cell division that occurs in cancer. I'd also like you to be able to compare and contrast two different types of genes that play a role in the development of cancer: oncogenes, or cancer-causing genes, and tumor suppressor genes, which, when they're normal, prevent cancer from occurring. And then I'd like you to explain the relationship of cancer to cell cycle control and how that gets disrupted. So when the cell cycle goes wrong, this leads to cancer. And what you can see over here on the left are a bunch of cancer cells growing and dividing under the microscope. These cells are called HeLa cells because they were isolated from a woman named Henrietta Lacks. And so these cells were taken from Henrietta Lacks's cervical tumor, a tumor that wound up killing her. And the doctor who took it, unbeknownst to her, sent a sample of those cells to the lab, to a research lab, and that research lab grew those cells, and it turns out that these cells continue to grow and reproduce indefinitely. These cells are what we refer to as immortal because they don't die when they're supposed to, and instead they just continue to grow. They don't have any cell cycle control or regulation left, and so they'll just continue to grow and divide even though they're not supposed to. However, although these cells were horrible for Henrietta Lacks and wound up killing her, they've completely revolutionized science, and how we've done science and all the things that we've learned from these cells is completely astonishing. You can read a lot more about it in this book, The Immortal Life of Henrietta Lacks by Rebecca Skloot. It talks both about the sociological and societal issues involved in this isolation. It tells you more about Henrietta Lacks herself, who was an amazingly strong woman, and it'll tell you more about the biology behind what these cells are. I really enjoyed this book, and I found it completely changed how I thought about cancer and raised a bunch of issues for me to think about medical treatment and patient information and confidentiality. So I highly recommend it if you haven't read it yet. So cancer, as you know, is a horribly detrimental illness. It's actually a collection of different diseases. Each different site, each different place that you could have cancer is its own completely distinct disease. And even within those, even within lung cancers, there are multiple subtypes of lung cancer, each of which behaves and responds differently to treatment. So as a result, cancer treatment is very, very challenging. These two different figures are showing you how important an issue cancer is, how important an issue cancer is nationally. So on this graph on the left, it's graphing the rate of cancer diagnoses per 100,000 individuals over the past 35 years, 40 years. And what you can see here is that cancer rates have fallen since the 1990s, but that they still are very different for people of different racial backgrounds. Black males have the highest incidence of cancer, and white females have the lowest incidence of cancer. On the right, you can see this information broken down a little bit more, and interestingly, it splits new cases, so new individuals diagnosed with cancer, and deaths, those individuals who die each year of cancer. And as you can see, white and black people have the highest rates and highest death rates of cancer, and Asian and Pacific Islanders and American Indians and Alaskan Natives have the lowest rates of cancer. So what is cancer? So cancer is a group of diseases, so multiple different diseases here, characterized by uncontrolled cell growth and the spread of abnormal cells. So these are cells that grow when they're not supposed to. They're not regulated by the cell cycle the way they should be, and they spread abnormally. So they start moving throughout the body when they're not supposed to. If the spread of this cancer is not controlled, it can result in death. So there are two different types of cancer, or two different types of tumors. Sorry, my fault. there are benign, non-cancerous tumors. So these tumors, these abnormal cell growths, don't spread, but they can eventually change and become malignant tumors, and then they become cancerous. So malignant tumors have the potential to spread to other parts of the body. Tumors originate from a single cell, and what normally happens is a cell grows and divides into two cells. That cell grows and divides into two new cells. And then if one of those two cells gets damaged, it'll either repair itself and revert to a normal healthy cell, or if it can't repair itself, it'll allow itself to go through this process of apoptosis, which is programmed cell death. Basically cell suicide. It'll kill itself if it can't fix the problem. Cancer cells, on the other hand, are supposed to fix themselves, but somehow they don't. So the cell and its offspring mutates, and these cells start to grow abnormally. So maybe it has a problem, like it doesn't do the appropriate checkpoint at the end of G2, and so its DNA isn't fully replicated, or something like that. So the cell's supposed to stop growing and it's not supposed to continue, but instead it does, and it divides. And now its daughter cells have not only that problem, but maybe they don't have the right amount of DNA, or maybe they have DNA errors or mutations. Those cells then grow and divide, and then grow and divide, and grow and divide. And eventually you're going to get a tumor, which is an overgrowth of cells that has no useful purpose. Often the immune system will come in and kill off these cells that it doesn't need and that are growing when they're not supposed to, but sometimes they don't get identified because they look exactly the same as other normal healthy cells until they start behaving really badly. So normally, cells that are growing in the body not only are regulated by their own cell cycle, but they're regulated by cues from cells around them, from their environment. And so normally mammalian cells are regulated by nutrient availability, different growth factors from the environment, and whether or not they're attached to some sort of surface or substratum that allows them to grow as a single layer of cells. So normally a cell put down on a surface will grow and divide until it just totally fills that surface. And then if you scrape and remove a few of those cells, the remaining cells will grow and just fill in that gap and replace them. This is called density-dependent inhibition. The ability to grow on a surface is referred to as anchorage dependence. So cancer cells don't have this internal regulation. They don't sense that the world is becoming too crowded, and they just continue to grow and pile up on top of each other, creating this big mass here. Also, they don't exhibit anchorage dependence. They don't need to be touching that bottom surface in order to grow. They're more than happy to grow on top of anything else and become completely dysregulated. So here's the example of a breast cancer tumor. Here are a few cells that have started to grow and divide on their own when they're not supposed to, and they create a small lump. Those guys will continue to grow and divide, and eventually they'll start taking over the area around them and taking up additional space, displacing other cells, and invading neighboring tissue. Eventually, these cancerous cells now can spread outside of the breast tissue. They start going into the lymph nodes and the blood vessels here, and then they can travel to other parts of the body where they can land on your lung or your kidney or your liver and start to grow and divide there when they're not supposed to. The radio program, the NPR program Radiolab, has a wonderful story about famous tumors, and they have a fascinating, absolutely amazing description of what happens in tumors and the different abilities that those tumors gain in the process of the mutations that occur as they are growing and dividing and becoming unregulated. And so if you follow this link, which I'll post also on our Blackboard site, and you listen to minutes 13 to 16, actually you can really listen to the whole podcast if you have time because it's wonderful, but if you don't, just listen to these 3 minutes and you'll be able to hear their discussion of these tumors developing the ability to spit acid and to be able to leave the environment that they're supposed to be in and go somewhere else. And it's accompanied by some great sound effects. So it's really fun. I recommend listening. Why do you think cancer primarily affects older people rather than young people? So take about 10-15 seconds, read through these answers, and press pause while you're here and think about which answer you want to select. When you're ready and made your choice, then continue the video. Okay, if you've made your choice, now's the time to go on. If you haven't, pause the video and make your choice. All right, so the right answer to this question is answer C. So cancer affects older people rather than young people because cancer develops after multiple mutations. So it takes lots of changes in the cell in order to have cancer occur, and these mutations have to accumulate. They take lots of time. So let's talk about the different types of genes that can get mutated. So cancer is a genetic disease, not really a heritable disease, not a hereditary disease, but genetic because it has a base in your DNA, and it has a base in genetic changes to your DNA. And so what happens in cancer is that you get an accumulation of genetic changes, accumulation of changes in your DNA, where at least 5 and possibly as many as 9 different genes get mutated and get damaged. So you don't really inherit cancer because you need to develop it yourself in your own body, and we don't pass it on to our offspring. But we can inherit a susceptibility of cancer. So you might have heard about this in relation to things like BRCA1 and BRCA2, which are breast cancer susceptibility genes. And these can lead to individuals who have them having a greater chance of getting cancer. Not that they're necessarily going to get it, but they just have a higher possibility of getting it. So a lot of these genes that get mutated in cancer are involved in regulating the cell cycle. And so it turns out BRCA2, one of these susceptibility genes for breast cancer, is involved in DNA repair. Without one of these copies of it, the DNA doesn't get repaired the way that it's supposed to, and that can lead to the accumulation of additional mutations, additional genetic changes. You can get mutations in 2 major different types of genes to lead to cancer. Proto-oncogenes, or are called oncogenes once they have a mutation. These genes, when they're normal, they promote cell division. So these are the genes that say, oh, great, everything is normal, let's run through the cell cycle and let's move on to the next phase. So they encode the proteins like cyclins that encourage the cell cycle to continue. When they get mutated, however, they might tell the cell cycle to continue even when it's not supposed to. Tumor suppressor genes, on the other hand, they normally act as the brakes. So when they're normal, they block cell division and prevent the cell from dividing when it's not supposed to. Let's talk about proto-oncogenes in a little bit more detail. So here you have the gene for a proto-oncogene. You have the promoter, and then you have the part that encodes the protein itself. And here's your normal protein. So normally, your protein promotes cell division. There are a couple different ways you could get mutations that would lead this gene to cause cancer, mutations in this gene to cause cancer. You could get a stronger promoter. Suddenly, your promoter gets so much better, DNA polymerase binds to it better, more and more and more protein gets made, and so now you have 3 times as much protein being made, and the cell thinks it's moving through the cell cycle faster when it's not supposed to be. Or you could get 2 copies of this gene. You're only supposed to have 1 on each chromosome, but maybe something happens and you get 2. Now you're making twice as much protein as you're supposed to. Or the third option is that you get a gene product that is hyperactive. Now you have the same amount of the protein, but the protein has this new activity, and it's saying, hey, divide faster, go faster, go faster, go faster, even when it's not supposed to be doing that. If any of these types of mutations occur, now you can have an oncogene. Now this protein here can lead to cancer. When before, normally when it was a proto-oncogene, it behaved normally. It did what it was supposed to do. The other type of protein that can get mutated in cancer is a tumor suppressor gene. So normally, tumor suppressor genes inhibit cell growth and inhibit cell division. So you make a normal gene and you say, hey, cell cycle, slow down, go a little slower. And it controls how the cell is going to divide. But let's say you mutated that protein so that it no longer worked, no longer did its job. Now it's no longer saying, hey, slow down, don't divide so fast. And the cells can continue to grow and divide and grow and divide when they're not supposed to. So this would be an example of destroying the brakes in your car. Suddenly you can't stop when you need to, and you just keep zooming down the street, crashing into things that you're not supposed to run into, skipping stop signs because you can't stop. In the cell, this would be like skipping cell cycle checkpoints. And so cell division is no longer under control. It's not regulated anymore. So normally it prevents cancerous growth. And two different ways it could do that is by maintaining the integrity of the genome, so repairing DNA proteins, serving as a checkpoint, or by inhibiting cell division as a checkpoint protein. So if you have a tumor suppressor gene, you need to mutate both copies of the gene. So even if you mutate one of them, you still have a functional copy. And this is referred to as the two-hit hypothesis. And this is part of the reason why it takes a longer time period to develop cancer, why older people develop cancer, because they have to accumulate these mutations in both copies of the gene. So here is somebody who has sporadic cancer. They're born with totally normal, totally functional tumor suppressor genes. And then at some point, maybe when they're like 35, in one of their cells, one of these tumor suppressor genes gets mutated. Everything is still totally fine. They're still going to grow. They're still going to behave normally. All of their cells are still going to cooperate. But eventually, that cell might just by chance accumulate a second mutation in that other copy of the tumor suppressor gene. And in that case, maybe this occurs when they're like 75 or 80. Now this cell no longer can repair DNA the way it's supposed to. And so all of the offspring of that cell accumulate all of these mutations, all of these changes in their DNA, and can eventually develop a tumor. The reason why sometimes cancer can be hereditary and why you can inherit a higher chance of getting cancer is because if you happen to be unlucky and be born with one mutated tumor suppressor gene, you're totally fine. You'd never know. Everything would be totally normal until one day, unluckily by chance, you got a mutation in one of your cells in the second copy of that tumor suppressor protein, that tumor suppressor gene. And then you could develop cancer. And this might happen to you at a much earlier age than it would happen to somebody who didn't have that chance, who didn't start with that original mutation. And so this is how cancer can become hereditary. It can give you a greater chance of getting it and a greater chance of getting it younger. All right, so let's say you get a cancer cell. Your tumor cell here has lost the ability to attach when it's supposed to. It's no longer regulating the cell cycle like it's supposed to, and now it escapes from the tumor and it escapes into the blood vessel. And so it's moving through the blood vessel and goes somewhere else, and then it accumulates in a secondary location. And so the vessels of your circulatory system provide a chance or a pipeline for the cancer cells to move to other places in the body. So cancer cells, by the time that they are fully cancerous and malignant and moving around the body when they're not supposed to, they have a tremendous number of cellular abnormalities, of chromosomal abnormalities. So this is a karyotype of a cancer cell. Do you remember when we were looking at a karyotype of a normal cell? You have two copies. In a normal cell, you have two copies of each chromosome. So 2 copies of chromosome 1, 2 copies of chromosome 2. And here, each of them should be the same color. So you should have 2 yellow chromosomes. And here you should have 2 reds, and here 2 purples, and here 2 blues. But what you can see in this cancer cell is that now you have 6 copies of chromosomes that have chromosome 1 information on them, these yellow bits of information. And they have pieces of a whole bunch of other chromosomes sort of all jammed together. And then chromosome 2, you have 4 of them, but they've got these little green bits on the end that look like they came from chromosome 10. And then chromosome 3 is purple, but this one's got some blue stuff that looks like it came from chromosome 15. So basically, chromosomes get all tangled and all shuffled, and you get the wrong number. Sometimes you get too many. Sometimes you get too few. Like here, you only have 1 copy of chromosome 21. It can be a really big mess by the time cancer is fully developed. If you do get cancer, there are 3 possible treatment mechanisms, and usually all 3 of them are used at the same time or in close period of time in order to try to eliminate the cancer as quickly as possible. So the first thing to do is to do surgery to remove the tumor. This will remove most of the abnormal cells, at least the ones that are in the original place that the tumor started to form, but it won't target some of the cells on the periphery of it or any cell that is metastasized and moved to any other part of the body. The second type of treatment is to do radiation. So radiation is targeted on that original site where the cancer cells are located, and it'll kill any additional cancer cells that weren't removed by the surgery. This is really important because you don't want those cancer cells, you don't want to miss any single one because you don't want those cancer cells to come back. The radiation will, however, damage some of the cells in the area, and that's just one of the unfortunate side effects of radiation. Chemotherapy, the third option, is systemic, so you have that throughout your entire body. And so chemotherapy is anti-cancer drugs that are used to kill cancer cells, and they're used to kill actively dividing, rapidly dividing cells. So one example of common types of chemotherapy drugs are ones that are used to block the mitotic spindle or other aspects of the cell division machinery. So if you block cell division, you'll block any cell that's in the midst of dividing. This is really good at targeting cancer cells because cancer cells are dividing very, very rapidly. However, it'll also divide any additional cell. It'll kill any additional cell that's in the midst of dividing. So as a result, this is going to target cells that divide pretty rapidly on their own. So things like skin cells that repair and replace pretty quickly. The cells that produce your hair in the hair follicles also grow and divide really rapidly. So this is why really dry skin and hair loss are common side effects for chemotherapy. The third really common side effect for chemotherapy is nausea or inability to eat and a loss of weight and things like that. And that's because all of the cells that line your gut, that line your digestive tract, also grow and divide really fast. And killing off those cells result in a decrease in the ability to absorb nutrients and generally making food and any digestive process really unpleasant. And so those are the common side effects of chemotherapy, and it makes perfect sense once you understand how those chemotherapy drugs work, in that they're killing any rapidly dividing cell, cancer cells, and then the side effects are for ones that are not cancerous.